very much, and thanks very much to the organizers for inviting me. It's a great privilege to be up here with so many great scientists. I'm going to talk a little bit about salmon. I guess I can go straight to this. So what are the salmonids? The salmonids are a group of fish, needless to say. They're the single family in the Salmoniformes. There's about 11 genera, and I think I have a here. So all of these are 11 genera. There's greater than 70 different species. All of the species are economically interesting, economically important, and I hope that I'll be able to show that they're all scientifically interesting. They are to me anyway. So what do we know about these fish that make them biologically interesting? We see that their common ancestor here was probably a freshwater fish, and in 2 times during their evolution, they have become anadromous. Anadromous means that they're born in freshwater, they go to the sea, then they return to their natal stream to spawn. We see down here the 5 species of Pacific salmon. They're classified as semoparous. That means they can only spawn once, and after having spawned, they die. Whereas the rest can have the capacity to be repeat spawners. If any of you have eaten salmon, and I hope you all have and will continue to do so, then you'll probably realize that the flesh has a pink color. Not all salmonids have a pink flesh, and we can see that the pink flesh is restricted to the salmon, trout, and char. So we can see that this trait evolved at this point during the evolution. However, what really makes the salmonids of prime interest to me and to many other people is that they're derived from a common ancestor which underwent a whole genome duplication. And that's illustrated by this arrow here. This whole genome duplication has influenced, I believe, not just the structure of the genome, but also the biology of this group of fish. Now, many people have said to me, we've got a fish genome. We've got a couple of fish genomes. Why do you want to sequence the salmon? And I have to point out to them that this group of fish is very far away from the, evolutionarily speaking, from the fish whose genomes have been sequenced. And they include danio, the zebrafish, cod, stickleback, medaka, and the pufferfish. And it would be like saying, well, we've got the chicken sequence. Why do you want to sequence a human? Why don't you just use the chicken as the reference genome? So what we want to do, of course, is to have a reference sequence. We've been working on this project, or projects related to this, for approximately 10 to 12 years. And then, in fact, I guess we can say quite crudely, we've done it ass backwards. But I was very pleased to hear the words of Eric Lander yesterday when he said, what made the Human Genome Project really worthwhile was the resources that were developed along the way. And so what we actually did was we developed the resources and then decided when we had the resources, it was time to actually try and get the sequence. So what have we done? We've got genetic maps. We've got an extensive EST database, over 500,000. We've got a physical map. These are integrated. They're integrated with a karyotype. We've got a big repeat database. And we've got a good knowledge about the genome. Now, compared to the wheat genome, I guess we have it easy. But we don't really think so, because it is a complicated genome. It's 3 gigabase, so it's human size. But it is very complicated in terms of the duplicated regions are greater than 90% identical. It's greater than 60% repetitive, and the repeats are long, anywhere from 1.5 to 3.5 kilobase pairs. We know a lot about the annotation. We've got over 18,000 full-length single-clone cDNA sequences. So we know a lot about many of the genes. We've sequenced more than 120 bacterial artificial chromosomes, so we've got a good snapshot of the genome. And we've assessed next-generation sequencing technologies as a means of going in and sequencing, and we found that most of them were really wanting. This was our coup de grace. Uni Grimholt produced Sally. So Sally was totally homozygous. So there's some things you can do with fish that you can't do with mammals. And we can produce totally homozygous fish. And by sequencing Sally, in her genome, then we overcome some of the problems that we would have of allelic variation, for example. We've decided to do the sequencing in 2 phases. This is probably the last major genome that was sequenced by traditional Sanger technology. So small shotgun libraries, BACs, and phosmids. We've got a 4 times coverage of that now. We've got over 100 times coverage of next generation sequencing using Illumina, both paired ends and mate pairs of different sized libraries. And I can tell you that practically none of that actually had a positive impact on the assembly of the genome. And we're now into other phases where we're using PacBio and other technologies. All of this is displayed in the white paper that we put out, and the first assembly was put into GenBank about a year ago. It contains about 560,000 contigs, and 50 contig size is about 9.5, and it covers 2.4 gigabase, so about 80% of the sequence. These are in contigs, not scaffolds. Okay, so obviously, the reason for having the salmon genome is to exploit it. First exploitation will be to study the rediploidization of duplicated genomes. This is the academic side of things, my particular interest. However, we will also use this to get basic information about the biology of salmon, their ecology, their physiology, their nutrition, the immunology. We're very keen on the management of salmon populations, wild populations. Salmon is an icon in British Columbia, where I come from. We also want to apply this, go back. We want to apply this to aquaculture, and we want to apply it to change over the feeding strategies, get new diets, for example, actually try to get salmon to become vegetarians without challenging the taste. But it's really sustainable aquaculture, wild fish conservation, and monitoring aquatic health. There's the rationale for doing the sequence. But what I want to talk to you today about is a little bit about some of the biology, interesting biology that's come out as we tried to apply genomics to a particular problem in aquaculture. So what happens with salmonids, or the model organisms? We're studying the result of a genome duplication. Ono, in his 1970 book, a book that has been referred to as often cited but seldom read, is really put forward the case for why evolution has really advanced as a result of gene and genome duplications. So we start off with a diploid, it becomes a tetraploid. This is probably highly unstable, and then it reverts back to a diploid state, and this is something we're particularly interested in. We know the ancestral karyotype was probably 2N of 48 acrocentric chromosomes. So the duplication, we now have 2N of 96 chromosome arms. And if you look at extant salmonids today, if we look at Atlantic salmon, for example, chromosome numbers can vary from 54 to 58. Not your normal sort of thing in a species. Chromosome arm number can vary from 72 to 74. The closest relative to Atlantic salmon are brown trout, 2N of 80. But the arm number can vary from 100 to 102. So some of the questions is, how does a genome achieve stability through diploidization? When does this happen? Does this cause speciation events? Is it different in different salmonid lineages? And in particular, how is sex determined? So we don't know the sex-determining gene in Atlantic salmon. But let's see what happens after a genome duplication. Let's assume it's an XY system. You end up with 2 X's, 2 Y's, a couple of autosomes here. Now, what's going to happen? Is one of the Y's going to say, I'm the master Y? Does one of the Y's get lost? Or does the genome say, enough of you, we're going to take a new transcription factor, for example, one of the new duplicates, and we're going to transform that so that it becomes a new sex-determining gene. And if that happened in different populations, the populations would possibly no longer be able to interbreed, and this too could cause different speciation events. And one of the predictions from this hypothesis is that different salmonid species should have different sex-determining genes. And that's something that we're exploring at the moment. Okay, so we know that sex is genetically determined in salmonids. The cytogenetic evidence for this. I'll show you some of the sex reversal evidence, and from genetic maps, we can see that there are sex-linked markers. Okay, application time, product, or try to. In Tasmania, they grow Atlantic salmon. Seems a little strange, but it works. And these Atlantic salmon actually come from eastern Canada, from the River Phillip. There were 3 introductions in the 1960s. They were then moved to Tasmania in the 1980s, and there's a breeding program there that we're involved with, and Saltas has that. So the problem is that where they grow the fish in the salt water, the water temperatures are rather warm. And so males mature very quickly. And as they mature, the flesh quality goes down, and they become more susceptible to disease. So that's the problem. What's the solution? The solution is to grow all-female stock. How do you do that? As you get fish eggs, as they're developing, you treat them with 17-alpha-methyl testosterone during sexual development, and they all become phenotypically male. But the pool is these neo-males, sex-reversed females, and true males. Then the trick is get rid of the true males, take the sex-reversed females, which are now neo-males and running milt, cross them with regular females, and you get all-female broodstock. If this misidentification occurs, then it's very costly, because now 50% of the offspring are fish that you're feeding, taking up tank space, but getting no return on investment. So our task, should we wish to accept it, was to actually identify markers that could discriminate between sex-reversed females and true males. We thought this would be a breeze, so we said yes, no problem. We had previously identified sex-determining locus in Atlantic salmon on chromosome 2. So we took microsatellite markers from chromosome 2. We took 7 families, which were derived from 4 males. We went in, did haplotype analysis in chromosome 2, and what we see here is these are sire 1, sire 2. These are the different haplotypes. M represents males, so we've got 12 males and 2 females in this particular family. And lo and behold, sex is not in chromosome 2 in these families. So where is it? That's the question. We did a little genome-wide scan, and using the genome-wide scan, we identified chromosome 6 as a candidate. Took microsatellite markers, do the haplotype analysis, and here we see the sire 1, sire 2 haplotypes. We go through the males and the females, and it's all right apart from male number 2. And this actually illustrates one of the big problems. It was a phenotypic miscall. So the genotyping is actually easy to do. The phenotyping, as we've heard, is actually very, very difficult, even for a simple thing like what is the sex, what is the gender of a fish. Anyway, we now have sex on chromosome 2. We go back to the mass pedigree, and we find that all of these 4 males that gave the 7 families are brothers. So they all come from one male lineage. So we said, oh, OK, let's go on. Let's look at some other families. So we now are looking at families using chromosome 6 markers. Lo and behold, no. Sex is not on chromosome 6 in these families. Where is it? It's on chromosome 2. OK, so you think, OK. We better check some others. So we've now looked at pretty much all of the lineages in the Tasmanian family, and we came across 2 families where sex was not linked neither to chromosome 2 nor chromosome 6. And we now know, cut a long story short, it's on chromosome 3. So what does this tell us? In summary, then, from 38 lineages, we've got 24 of these male lineages. The sex locus, sex meaning the male phenotype, is mapped to chromosome 2. In 12 instances, it's mapped to chromosome 6. And in 2 instances, it's mapped to chromosome 3. Are there more than 3 sex loci in Atlantic salmon? These all came from the same river. their founders came from one river in Nova Scotia. Maybe something going on in Nova Scotia. I don't know. Is it the same master sex determining gene, or are they different genes? So how do we know? What do we do here? Well, in June of this year, Jan Geegan's group in France identified SDY as the sex-determining gene in rainbow trout. That was good. It's a paralog of interferon regulatory factor 9. That was a total surprise, total shock. And this is interferon regulatory factor 9. It's a nice DNA binding domain, they think. Transcription factor, could be. SDY is lacking the DNA binding domain. It's not a traditional transcription factor. So what do we do? So is SDY also the sex-determining gene in Atlantic salmon? We designed primers. We isolated the gene. We designed primers. We do PCR tests. Here, none of the females have SDY. This is SDY exon 4 and exon 2. And this is fatty acid binding protein 6b, which is a positive control to make sure our DNA amplifies. The males that come from lineage chromosome 2 or 3 or 6 all have SDY. But we haven't mapped the SDY. So is SDY on chromosome 3, 2, 3, and 6, or is it on somewhere else, and we now have different sex-determining genes on these chromosomes? How do we try to address that? We do fish analysis. So we isolated a bacterial artificial chromosome that contains SDY, carry out the fish, and it's shown in the red markers, and it goes to 3 pairs of chromosomes. When we do dual fish with the green labels, we find that it goes to chromosome 2, chromosome 3, and chromosome 6. So we now know that the region round about the SDY is at least hybridizing to those regions, and so we're now trying to get a smaller probe that is only SDY and see if, in fact, this is what's going on. One of the surprises that we've seen in salmon genetics, genomics. And I don't know if there are many other instances where a single species has different sex-determining loci. The only one I'm aware of is in Arctic char, which is also a salmonid. That's the work of Roy Dansman's group. So what are the conclusions that we've learned so far? Atlantic salmon genome is large and complex, not as big as wheat by any means, but we think it's pretty complicated. We've much to learn about the genome, but how it's been sculpted as a result of the whole genome duplication. We're pretty sure that salmonid biology has also been influenced and shaped by this whole genome duplication. We believe that having the Atlantic salmon genome as a reference will allow resequencing of other salmonid species, and it will allow us to address some of the interesting biology, such as why do the Pacific salmon die after spawning, whereas other salmon don't, for example. And we expect that the understanding salmonid biology will allow us to apply this more to the aquaculture industry and the salmonid conservation biology industry as well, using hatcheries and the like, and we'll see what happens from there. So I'm just a spokesperson. Many, many people contributed to this work. Primarily, Will Eisbrenner did all of the genetic mapping, and Song Lin and Jiang Li did the fish analysis. Little note for those of you who want to get funding from Genome Canada, take Pierre fishing and make sure he catches a fish. Thank you all.